On this week's episode, we turn the mic over to a pair of talented researchers with Assistant Professor Travis Schlappi and PhD student Tochukwu Dubem Anyaduba. As they ask each other questions, this is a great opportunity to learn about their cutting-edge research and also hear Dubem describe his career aspirations.
All right. Hi, Travis. Can you describe how your research has evolved over the years?
Sure. So I'm a new faculty here at KGI. I've been here a year and a half. In my PhD, most of my research was on detection of various infectious diseases, HIV or hepatitis C, or these different viruses. And in the later parts of my PhD, and now that I'm at Keck Graduate Institute, my research has evolved more into not just detecting the disease, but also a diagnostic to help the doctor treat the disease and know which drug or antibiotic will effectively kill the infection. So instead of just detecting HIV, now we're working on UTIs, urinary tract infections, but we want to detect the urinary tract infection and then also take that next level of identifying which antibiotic or which drug will effectively kill the urinary tract infection. And this is important because antibiotic resistance is spreading. If you've seen it in the news, I don't know, Dubem, if you read the news, but it's all over the news. Superbugs, gonorrhea superbugs that are resistant to everything, or Enterobacteriaceae superbugs that are resistant. There's carbapenem resistance spreading all throughout the hospitals, and MRSA, which is methicillin resistance. So it's a really huge problem when the antibiotic that doctors typically use to kill the infection doesn't work anymore, because that's how life is and bacteria evolve resistances. And so it's not enough to just identify the bacterial that's causing an infection. We also need to identify what kind of resistance the bacteria has and which antibiotics it will be susceptible to, which antibiotics will actually work. So the research has it's taken a fun and interesting direction into antibiotic susceptibility testing, and I'm enjoying it a lot. It's a big scientific challenge and lots of technology research and development engineering going into it.
So, Travis, you did mention about antibiotic resistance and susceptibility. Would you care to explain more so that our listeners can understand, why it's so much important now for you to do this research?
Sure. I'd love to explain a little bit more about that. And so one of the biggest problems with antibiotic resistance is the patient health that it's affecting. And so the current treatment paradigm is a patient, their bladder's on fire, they think they have a UTI, a urinary tract infection, they go to the doctor, he prescribes an antibiotic, patient goes home, and most of the time the antibiotic works and the infection goes away, but its resistance is increasing, and patients then have to return to the doctor. And they say, hey, the antibiotic is not working. So then the doctor will prescribe another antibiotic and see if that one works. The problem with this treatment paradigm is that the doctors, the physicians, are guessing at which antibiotic will work, and it's an educated guess based on historical and epidemiological data, but they don't know for sure if the antibiotic is going to work or not yet, because currently it takes 2 to 3 days for current methods to determine antibiotic susceptibility. And so the patient doesn't want to wait 2 or 3 days. Their bladder's on fire, and so he or she, the physician, will prescribe an antibiotic immediately to relieve the pain. Now, this is a small example of a bigger problem. So urinary tract infections don't cause mortality very often, but this also happens in blood infections, diarrheal infections, and respiratory infections. Any kind of bacterial infection, we're running out of antibiotics to treat them with. And so if we can show a proof of principle concept of a diagnostic device, of a technology and method that will successfully identify the pathogen and the antimicrobial resistance, then we can apply it to any type of infection. There's estimates out there by experts in the field that antibiotic resistance, if we don't do anything, if kind of like global warming, it's not a huge problem now, but in 50 or 100 years, the world may overheat and it'll be a huge problem. Same thing with antibiotic resistance. If we don't stop it now, estimates are that by 2050, it will cause up to 10 million deaths worldwide. And just so you have a reference for that, cancer currently causes about 8.2 million deaths. And so that kind of puts it into perspective that, hey, if we don't do anything, antimicrobial resistance can become as big of a problem as cancer. And then there's the economic side of things. If patients stay in hospitals longer because their infection is not being treated well, then that increases healthcare costs. They stay longer. They're not at their jobs as long. There's more physician costs, treatment costs, drug costs, everything. And so the estimates are anywhere between $1 and $100 trillion of lost economic output, adding up all the years from now until 2050 if we don't do anything about antibiotic resistance. So that 1 to 100 trillion is a huge number. And to put that in perspective, the US GDP per year is about $16-17 trillion. So it can be a huge healthcare and economic problem if we don't do anything about it.
In your current research, how many people do you have in your team since joining the KGI?
So, yeah, our team is growing. I've been here a year and a half, and I'm happy to say we have several students that have joined. I have one PhD student, Dubem, and then there's 2 master students currently working in the lab. We have 5 more. So we have 2 master's thesis students and then 5 other master's students that are working for independent credit, kind of on the side. We actually had a pretty successful summer. We had some visiting undergraduate students from other colleges that came over the summer. And then we have a high school program, and we had 2 really good high school students that have made some significant contributions to the project. So it's fun to get a lot of people involved from different colleges and age groups, and especially here at KGI to have students actually actively applying what they learn in class into the research lab. And it's good for them to see the connections between what they learn in class and how it can help people.
What, in your opinion, is the unique thing about KGI?
So what I really like about KGI and what makes it unique, apart from pretty much any other university I've kind of gotten to know, is it's has a very large emphasis on the translational side of science and medicine. And translating science using business to translate science into products that people will actually buy and that will actually impact society. And so I don't want to bash on basic science. Basic science is important. And sometimes basic scientific discoveries, they do translate 20 or 30 years down the road into things that benefit society greatly. What I think is unique about KGI is there's a big emphasis, and not just emphasis, but there's resources, there's opportunities for faculty, staff, and students to take the discoveries that they make in the lab and to use the business resources and skills here at KGI to translate those discoveries into industry, to manufacture, whether it be devices or drugs or whatever it be, to actually make it impactful for society and not just a cool scientific discovery. So I'll flip it around on you and ask some questions. So Dubem, can you talk about what you're currently researching and how that fits into the research team's overall goal?
Thank you, Travis. The overall goal of my team basically is to develop medical devices and diagnostic assays that are fit for use in point-of-care settings, and perhaps being able to apply them in remote settings or resource-limited environments. So that's one of the key goals of our research team. And my current research right now is to develop assays or technologies that could be used for multipathogenic screening. What I mean by multipathogenic screening is to be able to diagnose or differentiate disease-causing organisms present in samples from diseases like patients, UTI patients, or patients suffering from maybe that has upper respiratory tract infections, maybe diarrheal diseases. So these diseases that I mentioned are caused by multiple organisms or multiple pathogens. So being able to characterize them and differentiate them to know the particular one that is responsible for the patient's ailment is very key to, as Travis mentioned, to antibiotic susceptibility. So that's one key part of my research, to develop those technologies, and also downstream to develop medical devices that will be able to work hand in hand with those assays that we are currently developing.
And so a follow-up question for you, Dubem, is something I failed to mention when I was talking about our research is, what is your goal for the time frame of these assays and devices that you're developing? How quickly do you want it to work? If it's at the point of care, as soon as you get the sample, how much time before you'll have the result?
Yes, so this is really key. In point of care settings, you want to have your results less than an hour, because most patients did not really have that luxury of staying in the hospital for a long time, and the doctors may only have like 45 minutes to attend to the patients. So you want about between 45 minutes to 1 hour to get results back to the patients. So the diagnostics we are currently developing to work within this time frame, so that on the first visit of the patient to the hospital or to point of care environment, they'll be able to receive not just the diagnostic, but also to be able to receive the right antibiotic for right drug for their ailment.
And as I was talking earlier, current methods usually take 2 or 3 days. So shortening that into 1 hour would be a huge improvement and enable the doctor to prescribe the correct antibiotic at the correct time without having to wait 2 or 3 days, and then in the meantime just guess at the right antibiotic. So another question for you, Dubem, is what are a few key things that you've learned from your time in my lab?
Yes, so one of the key things that KGI drives on is teamwork. So I spent a semester studying MBS program in the MBS program, and it emphasized a lot of teamwork. And coming to Travis Lab, I got to practicalize that, because I now have a team of students that I mentor and also train in the lab, and being able to work with them to figure out the next stages of experiments, design experiments together, or figure out what their training needs are, is very key. So I've learned a lot about team building and team working together as a team. That's really critical. And another thing I've learned so far, more importantly, is problem solving. So like I mentioned, one of the major downsides of current diagnostics is being able to differentiate multiple pathogens within a sample. A lot of diagnostic devices out there do it by differentiating the samples into multiple wells. These add to the cost and also complexity of the device. But what we are trying to do is to be able to solve that problem, to be able to have that diagnostic work within one sample, sample vessel. So being able to do that is a huge task on problem solving. And right now, what the technologies we are currently experimenting on are really creative and innovative, and being able to tackle the challenges we face day to day running these experiments really improves my strength in problem solving.
Thank you. And then finally, how does your current research tie into your career aspirations?
Thank you. My career goal primarily is to be able to contribute to global health research, and more importantly, contributing to democratized research in Africa. A lot of diseases we have in Africa, for example, in 2016, the World Health Organization showed statistics published a statistics to show that infectious diseases such as malaria, diarrheal diseases, lower respiratory infections are actually the top part of the top 10 in the causes of death in 2016. Now, this is challenging because that statistic is different for developed countries. One thing I want to do is to be able to get the skill, take the skills I gain here during my research back home and be able to contribute to the growing strength of researchers that are tackling the problems locally. It would be nice to tackle the problems from here, but being at the forefront of the research and being able to understand what the people need locally, and also we'll be able to drive the direction of research. So my career goal is to be part of that workforce that is currently growing, and more importantly, not just being able to develop the research or develop the current technologies that will be amenable to Africa, but also to be able to teach the younger growing generations. I wouldn't be fulfilled in my career pursuit or my professional pursuit if at the end of the day I'm a fulfilled scientist and I'm published, but then the younger generations don't learn anything from me. So I want the younger generations to be able to gain from my wealth of knowledge that I'll develop here. So for that reason, I have a dream of pursuing or continuing a career in academia.
Okay, thank you. Yeah, we'll wrap this up. I would like to express appreciation to all the members of my lab, both past and present, that have helped with this project. And for anyone interested in contributing to the research that we have going on here in the Schlappi lab at KGI, feel free to look up my bio with the link and send me an email.